So why did we choose to work with zebrafish? Well, the big advantage of zebrafish over mice is that all development is external. But there's a few other advantages as well, but there's also some, disadvantages. So another huge advantage is that they're very cheap to maintain, and they're an incredibly hardy animal. They tolerate a lot of temperatures, although their favorite temperature is about 28.5 degrees, and that's what we aim to achieve in the aquarium. That means that all the processes go on at the physiologically most favorable temperature. But you can slow down development by cooling the room, and you can speed it up within limits by warming up the room. So they can be housed quite densely, so we normally keep about 35 adult zebrafish in a 4-litre tank, whereas mice, generally speaking, you could only have 3 in a box. And you've got to keep the males away from each other because they tend to attack each other, whereas zebrafish are just really social organisms. They basically just go about their business, look forward to you dropping by twice a day with a bit of fish food, and they're just the easiest, most robust animals you could hope for. And another huge advantage from the science point of view is that a typical clutch, so that's the name we give to a litter, can easily comprise 200 to 300 embryos. So if you're studying genetics, and particularly in the context of mutants, where the bringing together of 2 recessive alleles from 2 parents that are heterozygous for a mutation means that the mutants that you're interested in occur at a ratio of 1/4. If you have a 200 size clutch, then 50 of the animals are going to be mutants. So that gives you loads of animals to work with. Whereas a typical litter from a mouse is 6 to 8, so you might get 1 or 2 mutants to study. And you can imagine if you then want to bring mutations together and generate the desired mutation 1 in 16th, it's virtually impossible for mice, where it's still easy to generate the animals you want with zebrafish. So the things we talk about are the external fertilization, the massive size of the clutches, the cheapness. But, you know, they're not as close to human beings as mice are. There's some fairly obvious things. So they have gills instead of lungs. Zebrafish in particular don't have a stomach, so you can't actually study anything to do with stomach development, or perhaps they're not a good model for gastric cancer. They don't have mammary glands, so not so good for breast cancer research. However, having said that, they do get cancer spontaneously in the wild, and it's very easy to induce either in the way that we do with mutations, or by exposing them to chemicals that are known to induce cancers in human beings as well. And when the zebrafish genome was completely sequenced and published a couple of years ago in Nature, it was demonstrated that about 85% of the genes in zebrafish and human were directly related to each other, and nearly all the genes that predispose to cancers are present in zebrafish, and that they therefore are capable of producing very valid models, not only of human cancer, but all sorts of other congenital and other diseases that occur over life. So we like to think the zebrafish and mouse models complement each other beautifully, and the interplay between the 2 organisms is, I think, together going to take us forward.